So we are starting to study cell structures by starting from the outside and kind of working our way in. Really what defines a cell, what makes a cell, is the presence of a barrier. All that a cell really is a compartment separating the inside from the outside. And when we draw a cell, when we look at what defines a cell, it's really the presence of this cell membrane. So we're going to talk about both the structure and the function. How is a cell membrane built, and how does a cell membrane work? So today we're going to start really by looking at how a cell membrane is built. What we're really doing here is taking the biochemistry that we learned and applying it to biology, applying it to life. So when we look at this picture of the cell membrane, we can call it the cell membrane, right? That's probably what you've heard it referred to. That's what it's referred to most often. It's also called the plasma membrane because it separates the internal plasm, the cytoplasm of the cell from the outside. But the name that tells us a lot more is the fluid mosaic model. Fluid mosaic model. So anytime you see cell membrane or plasma membrane or fluid mosaic model, they all really relate to the same exact thing. So this is, of course, a diagram, a drawing. You can use an electron microscope to get more of a photograph of what a cell membrane would look like. But this is a pretty important drawing because it's what we call space filling. It shows us the space that these molecules actually take up. So what we want to do is look at kind of the main components, which are just below the screen here, and then look at how they join together. So one of the biggest things is being able to notice that we kind of have this barrier or separation, and this is going to be selectively permeable. Selectively permeable. And we're going to use this phrase a lot during this section on cell membrane when we start to look at cell transport. The main idea that we want to know is that this barrier is not impermeable, meaning things can get through this barrier. And we've actually already talked about some of the main ideas of what might be able to get into a cell and why. Because remember, what all life is influenced by is chemistry. And so if these are just chemicals, then they're going to follow chemical behaviors. So this is why you needed to know what polar and nonpolar meant. This is why you needed to understand charge. This is why you need to understand that some ions or atoms are bigger or smaller than others. So hydrogen is the tiniest atom, and then we've got larger atoms. And so we need to know that because they're going to follow certain behaviors. So let's take a second and just kind of break this down to the smallest units. Really what the cell membrane is made of is the phospholipid, and that is the main component. So here we've got the phospholipid. This is going to be the main component. And we have already talked a lot about the structure of the phospholipid. Keeping in mind that we have this glycerol, this 3-carbon backbone. It's just a lipid. It's a 3-carbon backbone. And so we've got 3 places to attach. Here we've got 2 fatty acid tails. And we've got a phosphate group. And so, of course, what we know about the phosphate group, the PO4, is that it's negative. And so we've got a charged group up here. Fatty acids are mostly hydrocarbons, and so these are going to be very nonpolar. The other thing, the reason I like this other diagram as well, is that it's also showing this idea of kinks. So one of these would be saturated, one of these would be unsaturated. So taking a second to think about that. Remembering that when it is saturated, like this one on the right is, all of the carbons are bonded to hydrogens. Any available carbon bond is attached to a hydrogen. Over here, we're probably going to have a carbon-to-carbon double bond. That's going to cause the kink. That is going to increase fluidity. So one of the things that we need to really be aware of is that the structure of the phospholipid itself is variable, especially looking at these tails, and are they saturated or are they unsaturated? So when we examine the phospholipid bilayer, that's actually another word for the cell membrane, so I guess that's 4, phospholipid bilayer, we'll see that the tails of the phospholipids are variable. So this is going to be important because this is actually a space for regulation. This is an opportunity for the cell to control or regulate how fluid the cell membrane is. And a lot of this is going to be in response to temperature. If it's really hot, we might want it to be less fluid and stay a little closer together. If it's really cold, we might want to increase the fluidity. Same thing depending on the function of the cell. So different cells in the body actually have different membrane fluidity. So this is our main component, the phospholipid, and we need to know the structure and the behavior of this molecule. The next main thing that we have is going to be an example of a steroid called cholesterol. And cholesterol is going to be considered a lipid. It is going to be mostly nonpolar. This only tiny little exception over here, see this OH? Remember that functional group? OH is going to be hydroxyl. And that's going to be a little polar. But just a little tiny bit. There's just one hydroxyl group on the whole rest of it is going to be hydrocarbon. So we're going to be mostly nonpolar. It's almost like a little mini phospholipid. So an even smaller version. The advantage of this polar bit is that it is going to be able to have some part of it on the exterior. So when we talk about phospholipids, the important thing to know is that the arrangement of phospholipids is actually spontaneous. We take a bunch of phospholipids individually and just mix them together in water, then we're actually going to form a phospholipid bilayer all on its own. So we want to know that, and so maybe we'll write over on this side. This bilayer forms spontaneously. Sorry, that's spelling. Forms spontaneously because all life is surrounded by water. So if this is aquatic or water-based, and if up here is aquatic, then these polar heads, which are hydrophilic, these polar heads that are hydrophilic, are going to line up facing the water, and the tails are going to orient away, that idea of hydrophobic exclusion. So the creation of the cell membrane is mostly spontaneous. We don't have to put each piece in place. That as a phospholipid is formed, it kind of is attracted or repulsed or repelled by basic chemical formula behaviors. And so what we see is the spontaneous bilayer forming with the tails or the hydrophobic regions moving away from the water. As these are formed, the cholesterol is also going to have a very similar principle of behavior in that it's actually going to have this slight hydroxyl group facing the water and the rest of it facing the inside. We already talked about this a little bit in that this is going to increase fluidity. Noticing that it doesn't go all the way through both sides of the bilayer. It just fits into one side of the bilayer, and so what that means is that it's just going to kind of push these two a little further apart, but the ones on the other side are still the normal spacing from each other. So by kind of squeezing in there, we're actually creating more space and making it more fluid, so there's more room for it to move around. The third major component is actually going to be proteins, and hopefully you recognize the alpha helix here in this picture. And you could talk about the 4 levels of protein folding, the amino acids in a sequence, and then the interactions between the main amino acid components themselves, those hydrogen bonding, and then the R groups, and then here it looks like we only have 1 subunit. But what we're going to find are proteins that are what we call embedded, meaning they are actually in the cell membrane. And those embedded proteins can be integral or peripheral, and we'll talk about those in just a second. The main idea meaning that they are in the cell membrane, and so that leads you to think for just a second. Think about what that would mean about amino acid sequence. If it's embedded into a cell membrane, we should really be able to identify what that means about the sequence of amino acids. And these proteins are going to have lots of functions that we'll look at in just a second. One other thing that we really want to add is this idea of we do have carbohydrates. And these are going to mainly, as we already discovered in class, function as ID tags. We're going to have different length of chains. We're going to have some that are straight, some that are branched, and those are going to help identify the cells within your body. So we're going to focus on the function a little more later, and now we're going to kind of combine the idea of structure versus function, or structure and function. And so what we have here are 6 different jobs that proteins can do in the cell membrane. So keeping in mind that the cell membrane is there to be a barrier, to separate the inside from the outside. There are going to be lots of things that that cell membrane also aids in. So what we have here is the internal environment. The yellow pictures are showing us internal, the blue are showing us external, and this is our selectively permeable membrane. All the different names we have for it. And so the very first job that we're going to see that the protein might have is it might be able to transport. We're going to talk about lots of different types of transport. We'll talk about facilitated diffusion. We'll talk about passive channels. We'll talk about active transport. But proteins that are embedded in the cell membrane can act as like an elevator or a channel or a route. They don't have to squeeze through the phospholipids. They can move through this protein, keeping in mind that proteins can have a specific shape and charge, depending on the amino acids. So we can actually control, I like this diagram because it shows only these red ones making it through. And so we can see that this channel is actually selective. The second function that these embedded proteins can have is actually to act as enzymes. They can catalyze a reaction. Of course, a specific reaction, but we will see that, especially when we get to cellular respiration, that we'll have proteins that are embedded in the cell membrane that facilitate or catalyze a reaction, so changing a reactant into a product. A third job that could happen is it could be a receptor. It can receive a signal, and it can have an action on the inside of the cell. So we talked about this a little earlier as well, but some signals will actually pass through the cell membrane if they're nonpolar, if they're small enough to get in. Others will actually have to bind to a receptor and then cause a change, keeping in mind that these receptors are also really going to be shape specific. So we see that theme, that pattern happening again and again. A fourth job that could happen is the possibility for identifying different types of cells. And so sometimes it's just going to be the presence of the protein that identifies. Sometimes we're going to see these additions of these glycos. And so these ones that are attached to proteins are called glycoproteins. The ones that are attached to lipids are actually called glycolipids. So we'll go more into this idea of identification a little bit later, but just that the cell membrane, since it's the barrier on the outside, we can actually identify the cells on the outside. Number 5 is what's going to be called adhesion, which is helping cells